Today we're going to be looking at muscles and muscle tissue. So first let's do a quick review. There are three types of muscle tissue: skeletal, cardiac, and smooth, and both skeletal and cardiac muscle have striations. Smooth muscle does not have striations. Skeletal muscle also voluntarily contracts, whereas cardiac and smooth muscle involuntarily contract. We're going to look at the structure, location, and functions, as well as the means of activation for these muscle types. And in all its forms, muscle tissue makes up nearly half of the body's mass. So let's look at these images first. And we have up in the upper left, we have a muscle type that has many nuclei, is long and cylindrical in cell shape, and has striations. So if you're guessing that this muscle type is skeletal, you are correct. On the right side, we have cells that are long and cylindrical, but have these specialized junctions called intercalated discs. We have less nuclei, so it appears the cells are uninucleate, and even though the cells are long and cylindrical, there is branching that can be seen. So this cell type is cardiac muscle, meaning down on the bottom is smooth muscle, which is also uninucleate, but has spindle-shaped cells, which are slightly fatter in the middle than they are on the ends. As we move forward, keep in mind there are muscle similarities. All muscles can contract because they contain actin and myosin. Actin and myosin are myofilaments, or protein filaments found within individual cells of muscle, and it is the reason, again, that cells that make up muscle tissue can contract. So we'll learn more about both of them. Prefixes like myo, mys, and sarco all refer to muscle. That means we have new terms like sarcolemma, which means plasma membrane of a muscle cell, or sarcoplasm, which means muscle cell cytoplasm. We can even call skeletal and smooth muscle cells muscle fibers because they do not branch. So we wouldn't call cardiac muscle cells muscle fibers because there is branching apparent in that type of muscle tissue. Let's take a closer look at skeletal muscle tissue. Skeletal muscle is attached to the skeleton. It has striations and it contracts voluntarily. So those are three words you should associate with skeletal muscle: skeletal, striated, and voluntary. We find skeletal muscle packaged around the skeleton, so it covers the bones, and it has obvious striations because of the repetition and pattern of the myosin and actin filaments. So said another way, the reason that there are striations in skeletal muscle and also cardiac muscle are because there are repeating and alternating patterns of the myofilaments in that type of muscle, specifically in that muscle cell. We also know that skeletal muscle is controlled voluntarily, so it's under conscious control by the central nervous system, specifically the somatic nervous system. Skeletal muscle can also undergo fatigue. That's because it contracts rapidly but tires easily, and it's responsible for overall body motility by attaching to bones, but specifically attaching to bones across joints. That allows not only for joint stabilization, but for us to maintain posture. And skeletal muscle contraction also generates heat because the chemical reactions that occur in skeletal muscles for contraction are not 100% efficient and therefore produce heat as a byproduct. Skeletal muscle can also exert just a little bit of force or forces of over a 70-pound amount. And that is because there are different motor units and you can use more or less motor units. And if you were to pick up a penny, you don't need to use as many motor units for skeletal muscle as you would if you were picking up a large, heavy barbell. Next, let's look at cardiac muscle tissue. It is cardiac, striated, and involuntary, meaning that it's found in the heart, contains striations, and contracts involuntarily, so it's not under conscious control. In fact, there is some automaticity, or contraction by itself, that we see in or from cardiac muscle tissue. This automaticity is controlled by the pacemaker cells that you learn about in Anatomy and Physiology 2, which sets the sinus rhythm of heart contraction at about 75 beats per minute. Now again, we have striations in cardiac muscle like we did with skeletal muscle, and the reason we have these striations again is because of repeating patterns of myofilaments. Again, those myofilaments are actin and myosin. Keep in mind also that cardiac muscle tissue is only found in the heart, not blood vessels. Even though the two parts of the cardiovascular system is the heart and blood vessels, the heart contains cardiac muscle tissue, whereas the blood vessels contain smooth muscle tissue. There are neural controls that are going to make sure that the body responds appropriately and contracts cardiac muscle involuntarily, and the neural controls for cardiac muscle and smooth muscle are the autonomic nervous system. And autonomic sounds like automatic, and that should help you remember that it's the autonomic nervous system that are the neural controls for cardiac and smooth muscle, which both contract involuntarily. That is as opposed to skeletal muscle, which was under somatic nervous system control. So soma, which is the first part of somatic, means body. So the muscle that moves the body is under somatic nervous system control. And we'll review that again. Also keep in mind that cardiac muscle tissue contains intercalated discs between their cells. Intercalated discs are special cell junctions, and those cell junctions contain desmosomes and gap junctions. Next, let's look at smooth muscle. Smooth muscle tissue is visceral, so associated with body organs, not striated, and is under involuntarily control. So it is also controlled by the autonomic nervous system. We find smooth muscle in the walls of hollow organs, so that includes blood vessels, many digestive organs, and urinary organs like the bladder. And we find that smooth muscle tissue is able to move substances along these hollow channels from one place to another, or from point A to point B, such as through the digestive system, where we see food forced and secretions forced through internal body channels. Smooth muscle's name tells you that it's not striated, so it does contain actin and myosin, but not in repeating, alternating patterns. So without that repetition of the myofilaments, we don't have sarcomeres, which are units of contraction. So since we don't have sarcomeres, we don't have striation. And more on sarcomeres, so I won't just introduce that term and not explain it, but it's coming up. So keep in mind that when you think about smooth muscle, you should think visceral, because it's found in the walls of hollow organs, non-striated, hence its name, and involuntary contraction, along with cardiac muscle, meaning they are both under autonomic nervous system control. There's some special characteristics of muscle tissue, so again, we're kind of looking at similarities, but then also pointing out the differences, because depending on which muscle tissue type we're looking at, it might be more or less excitable. But all muscle tissue is excitable. It's responsive. It has the ability not only to receive a stimulus, but to respond to that stimulus. And a lot of times that response is contraction. The response usually requires a generation of an electrical impulse that is traveled either from a neuron or created by the automatic pacemaker cells, say in cardiac muscle. But it's going to be an electrical impulse that travels along the entire plasma membrane of a muscle cell and causes that cell to contract. And this contraction signal can then travel to other cells that are attached to the one that has been electrically stimulated, and that would allow the electrical signal to travel throughout all tissue and therefore cause muscle as an organ to contract. So all muscle tissue is excitable. All muscle tissue can also contract, which makes sense because that's one of the main functions of muscle tissue is contractility, whether to move food along an internal channel or to pump blood throughout the circular system, or, for example, to allow for the body to move. So all muscle tissue can contract. There's also extensibility. Not only can muscle cells shorten, they can also stretch, they can be extended. So there's a fair bit of elasticity to muscle tissue as well, because after they extend, they can recoil to their original shape or their resting length. So let's see if we can match these muscle functions that are a little more specific than just contraction to the different types of muscle tissue. So I'll give you a minute to look through them, and then we'll review the answers. Okay, so muscles that are responsible for all locomotion. You should have said skeletal muscle. How about the second one? Muscle that is responsible for coursing the blood through the body. You should have said cardiac muscle, because cardiac muscle is in the heart, and the heart is a pump. How about muscle that helps maintain blood pressure and squeezes or propels substances through organs? You should have said smooth muscle. And that brings us to skeletal muscle again, because we are going to use skeletal muscle as our model for both the anatomy and later the physiology. So we'll be talking about skeletal muscle cells as individual units, but then we'll also talk about how skeletal muscle cells are organized to create what we call a skeletal muscle, which is an organ. So first, let's talk kind of gross anatomy. If we're looking at skeletal muscle, there are attachments to skeletal muscle that span joints. So most skeletal muscles span joints. That means they attach across joints, and that means that when they contract, they move a joint. There are two locations where skeletal muscles attach: the insertion and the origin. The insertion is the attachment of a skeletal muscle to the movable bone, whereas the origin is the attachment of the skeletal muscle on the immovable or less movable bone. A great example to remember insertion or origin is to think of a bicep curl. Your bicep brachii is attached to your radius and your scapula. When you do a bicep curl, your forearm moves, but your shoulder blade either doesn't move or moves less, meaning that the attachment of the bicep brachii muscle to the scapula is its origin, and the insertion for the biceps brachii muscle is on the radius. Also notice that the forearm or radius, when we bend the elbow, because the bicep brachii muscle contracts, is moving towards the scapula. So the insertion tends to move towards the origin when joints move due to muscle contraction. So the muscle's insertion moves toward the immovable bone, or the muscle's origin. Muscles can attach with direct or fleshy attachments or indirect attachments. We're going to learn what the epimysium is, but a direct attachment is when the epimysium of muscle is fused to the periosteum of bone. And I'll give you a quick overview of what the epimysium is. Epimysium is a connective tissue coat over muscle as an organ. And remember back to chapter 6, the periosteum is a connective tissue coat over bone. So essentially, the direct attachments are one connective tissue overcoat to another connective tissue overcoat, and it's muscle to bone. Then the indirect attachments are when we have the connective tissue wrappings. The overcoats extend a little bit further past the muscle itself and become a tendon or an aponeurosis, and then attach to the periosteum of the skeletal element or bone. So these connective tissue wrappings, again, could be a tendon or an aponeurosis. Now you're probably more familiar with what a tendon would be, so it's connective tissue at the end of bone, at the end of muscles that attaches to bone. An aponeurosis is essentially the same thing, but it's a flat tendon. For example, your frontalis, which is the muscle for your forehead, and your occipitalis, which is the muscle on the back of your head, they are attached via an aponeurosis. So that aponeurosis is going to go from the frontalis to the occipitalis, and because of the shape of your skull, it's a flattened sheet of connective tissue. So that would be an example of an indirect attachment between two muscles, but there's also indirect attachments between muscle and bone. There is a nerve and blood supply for skeletal muscle, meaning that it is vascular and innervated. Each muscle is actually served by one nerve, an artery, and one or more veins. Then each skeletal muscle fiber, remember that's a way we can talk about individual cells, has its own nerve, and that nerve ending controls contraction. That nerve ending actually gets really close to the skeletal muscle fiber, and we'll learn that that's called the neuromuscular junction, where a neuron and muscle meet. There's also contracting fibers that require continuous delivery of oxygen and nutrients via arteries, hence why skeletal muscle would need such a rich blood supply. You actually need a lot of energy, therefore a lot of oxygen and nutrients, in order for muscle to contract. And then wastes are always removed via veins. So in Anatomy and Physiology 2, you'll look more at the blood supply, at the cardiovascular system, at the circulatory system, and you'll see that arteries bring nutrients, veins remove nutrients or waste. Depending on what's happening or what type of tissue you're looking at, sometimes veins are used, for example, in the digestive system to remove nutrients from what we just absorbed and send it to the rest of the body. Alright, now let's get into the true gross anatomy of skeletal muscle. We have some basics. So each muscle is considered a discrete organ. It's composed of connective tissue, muscle tissue, blood vessels, nerve fibers, and we can arrange skeletal muscle as an organ using 3 connective tissue sheaths. And we're mentioning these sheaths from internal to external. So let's look at their prefixes. Endo in endomysium means inside or within. Peri for perimysium means surrounding or around. And we actually see that the perimysium is around or goes around fascicles. Then epimysium means on top of or kind of as an overcoat. And that's exactly what the epimysium is. So again, these are connective tissue coverings, and they cover certain things in muscle that are organized in a certain way to create skeletal muscle as an organ. And we'll also see some tendons or aponeuroses that are going to be there to attach muscle to bone or another muscle. Alright, so first let's talk about what the endomysium is. The endomysium is a really thin piece of connective tissue that covers each muscle cell. That's all it is. It's a thin piece of connective tissue that covers each muscle cell. Then we bundle all of our muscle cells together. And remember, they each have their own endomysium. And when we bundle muscle cells together, that's called a fascicle. And to hold the bundle of cells together, we have to wrap them with another connective tissue, and that's the perimysium. So the perimysium is just fibrous, a little bit thicker, but fibrous connective tissue that surrounds a group of muscle fibers, which is the same thing as muscle cells. Then, if we take all of our fascicles and bundle them together, to hold them together, we need another piece of connective tissue. And that piece of connective tissue is going to be on the outside. It's called the epimysium. This is an overcoat of dense regular connective tissue, similar to the periosteum in structure, and it just surrounds the entire muscle. So here are those tissues. So let's just point out what's here. We have bone, a tendon, and we have muscle as an organ. Let's start from the outside and work our way in. So what covers the outside of muscle as an organ is a connective tissue sheet called the epimysium. The epimysium is holding together a bundle of fascicles. Each fascicle, so they've pulled one fascicle out for us, is covered with a connective tissue sheet called the perimysium. And a fascicle covered in perimysium contains muscle cells. So they pulled out one of the muscle cells for us, also called a muscle fiber, and every single muscle cell also is covered by a connective tissue sheet, and that's the endomysium. So you can see if we look at a cross-section of a fascicle that each muscle cell, which is shown here as a red dot, has a white line around it. That's the endomysium. Then we bundled the cells together, each with its own endomysium, and covered it with another connective tissue sheet, and that is the perimysium. Then we bundled all of our bundles, which are fascicles, and we covered it with epimysium, which is the overcoat. Now I can also explain this organization of connective tissue sheets as bundles upon bundles. But I can also use inanimate objects to help you better understand this organization. So let's pretend that our muscle fiber, our muscle cell, is a straw. So let's start on the most internal or deep structure, which is the muscle fiber. Keep in mind, we're looking at skeletal muscle as an organ. We're not looking at individual cells. So skeletal muscle as an organ, meaning we're looking at gross anatomy. And we're starting with a straw. The straw is representing our muscle cell. So if I have my straw and I wrap it with a tissue, so I wrap a tissue around my straw, then what I'm demonstrating is a muscle cell wrapped in endomysium. And I'm going to do that to a bunch of straws. I'm going to have all these straws wrapped in tissues. but that's not very organized. So I'm going to bundle my wrapped straws together, and to hold them together in a bundle, I'm going to wrap them with saran wrap. Now my bundle is a fascicle. The saran wrap is the perimycium. And I don't bundle all of my cells together in one fascicle, because maybe I have 100 straws wrapped in endomycium or wrapped in a tissue. Instead, I put 10 in a bundle, wrap them with saran wrap. I put 10 more straws in a bundle, wrap them with saran wrap, and I keep going till all of my 100 straws are not only wrapped in tissue, but now wrapped in saran wrap. So I have plenty of fascicles. I'd actually have about 10 fascicles if I had 100 cells. Well, now I just have all these saran wrap bundles of straws laying around. So to organize them a little bit better, I'm going to bundle them together. So I take my saran wrapped, tissue wrapped straws, and I group them together. So I have 10 bundles, 10 fascicles. And now I'm going to take a blanket and I'm going to wrap this around my bundles of straws. The blanket represents the epimycium. So let's just do that example again. If I had a straw as my muscle cell and I wrapped it in a tissue, that would be my endomycium. If I took all my tissue wrapped straws and put them in a bundle, that would be a fascicle. And to organize my fascicle, I will wrap it in saran wrap, which is the perimycium. If I have a bunch of bundles and I bundle them together, then I have straws wrapped in tissue, bundled together, wrapped in saran wrap, and now I'm going to wrap all those bundles in a blanket. The blanket represents the epimycium. So bundles upon bundles is a great way to think of skeletal muscle as an organ. Again, from the inside out, the 3 connective tissue coverings that cover skeletal muscle as an organ are endomycium, perimycium, and epimycium. Endomycium surrounds each cell, perimycium surrounds each fascicle, which is a bundle of cells, and the epimycium surrounds the entire muscle as an overcoat, and it's going to be the largest or most external of the 3 connective tissue sheets. Now let's look at the microscopic anatomy. To look at the microscopic anatomy, we're looking at just one single skeletal muscle cell, one single skeletal muscle fiber. We know that a skeletal muscle cell, which we can call a skeletal muscle fiber, is long and cylindrical, it has many nuclei, and it's striated. The fibers themselves are 10 to 100 micrometers in diameter, and that's up to hundreds of centimeters long. The sarcoplasm, remember that word, that's the cytoplasm of a muscle cell. The sarcoplasm in a skeletal muscle fiber even contains myoglobin, which can hold on to the oxygen delivered to it via arteries. Because again, it's really important that muscle fibers have oxygen because they need it to make lots of ATP, which is cellular energy. Other things we're going to see in a skeletal muscle fiber are myofibrils, which contain myofilaments. The sarcoplasmic reticulum, which is just an endoplasmic reticulum with a special structure. So it's related only to muscle, so we're calling it the sarcoplasmic reticulum using one of those prefixes we learned. And T-tubules, which are a specialized structure that are continuous with the sarcolemma or plasma membrane. So let me review those again. Myofibrils, they are rod-shaped proteins that contain actin and myosin, which are the myofilaments. The sarcoplasmic reticulum is a specialized endoplasmic reticulum. And T-tubules are structures we see in specifically skeletal muscles and their cells, and they are continuous with the sarcolemma, meaning that the plasma membrane of a skeletal muscle fiber and T-tubules are connected. So let's just kind of review that terminology. This is kind of a cheat sheet for skeletal muscle. So sarcolemma, plasma membrane of a muscle fiber or cell. Sarcoplasm, cytoplasm of a muscle cell. Myofibrils, rod-like protein fibers that run parallel to the length of muscle fibers or cells. Myofibrils we're going to look at in more detail. They make up most of skeletal muscle cell volume, about 80%, and they contain the smallest contractile element of skeletal muscle called the sarcomere. Speaking of sarcomeres, they are the regular arrangement of myofibrils that account for the striations in skeletal and cardiac muscle. Again, they are the smallest unit of muscle contraction. They contain thick and thin filaments. That means that it makes sense for sarcomeres, which contain thick and thin filaments, to be found in myofibrils, which are rod-like proteins that also contain thick and thin filaments. So sarcomeres are just smaller units of myofibrils. The sarcoplasmic reticulum is modified endoplasmic reticulum, still interconnecting tubules. They surround each myofibril, so that accounts for their modified structure. And the modified ER, or sarcoplasmic reticulum, comes into contact with T tubules. The location where the T tubules or transverse tubules touch the sarcoplasmic reticulum are called a triad. There's two structures of the sarcoplasmic reticulum called terminal cisterns or terminal cisternae, and these are the structures that touch the T tubule. T tubules are also called transverse tubules, and they are also going to be interconnecting kind of flat tubes, and they connect to the plasma membrane or sarcolemma. But they are continuous with the sarcolemma on the outside, but contain or continue into the inside of the cell. So that means that anything that starts like an electrical signal in the sarcolemma can continue into the cell or into the sarcoplasm thanks to the T tubules. And the T tubules and the myofibrils are both going to be important in contraction. So I'll stop for just a moment and let you guys just look at this page. It's a great terminology page. It's good to make sure you know all of these terms before we continue forward. So if you need to pause this video here, please do so. Okay, so here is our microscopic structure. And I'm just going to go through and point out things we just learned about. Alright, first and foremost, we have the plasma membrane. For a muscle cell, that's called the sarcolemma, and they've pulled back part of it so we can see internal structures. But this clear structure that goes around the cell is the sarcolemma. Inside the cell, even though there's not a lot of space for the cytosol and the organelles, everything inside is the sarcoplasm. We have mitochondria, there's some ribosomes in here somewhere, but the big structure you're seeing that you maybe haven't seen before inside of a cell is the rod-shaped structures, one of which they pulled out for us. These rod-shaped structures are myofibrils. Myofibrils contain myofilaments, and parts of the myofibril are called sarcomeres. So I'll point that out again. So we have a skeletal muscle cell covered with a cell membrane. Inside is the cytoplasm, which contains organelles and cytosol. One such organelle is a mitochondria, but it also includes, for skeletal muscle, myofibrils and things like T tubules in green or the endoplasmic reticulum in blue, even though it's modified, so it's called the sarcoplasmic reticulum. So we have all these structures inside the cell, that's the sarcoplasm. If we pay attention first to these rod-shaped structures, we can note first that they're called myofibrils, and then that inside the myofibrils are actin and myosin, because those are the myofilaments. The myofilaments are also sometimes called the thick and thin filaments, the thick filament being myosin, the thin filament being actin. So you can see they've even displayed that here with a slightly thicker yellow line compared to the dark red. Also notice that we have these zigzag structures, one zigzag to another, so we can't see where the other zigzag would be, but one zigzag to another would be a sarcomere. So parts of a myofibril, certain links of a myofibril are sarcomeres. And if you have repeating sarcomeres, that's going to be enough to make up a myofibril. For example, we see one zigzag line here, here's another. So this length, this portion of a myofibril is a sarcomere. And you'd have repeating sarcomeres making up these long myofibrils, meaning that since sarcomeres are units of a myofibril, they both contain actin and myosin. And let's just look at this green structure. This is a T-tubule. Notice that there's areas where the T-tubule has folded inwards, and that is because the T-tubules actually start at the sarcolemma and then fold in, and that's why they're connected to the plasma membrane or sarcolemma of a muscle cell. So again, anything like an electrical signal that reaches the sarcolemma can go into the cell because of the T-tubules, because they're connected. The T-tubules are continuous with the sarcolemma. Also notice that there are two blue structures that touch the T-tubule. Those two blue structures are terminal cisternae, or just end parts of the sarcoplasmic reticulum. And since we have two terminal cisternae, because there's a blue part of the sarcoplasmic reticulum that touches each side of the T-tubule, the whole structure is called a triad, because if you count one terminal cisternae plus one T-tubule plus another terminal cisternae, there's three structures. So it's not complicated when they call it a triad, they just counted how many structures touch. And of course there's also a nucleus. There's plenty of nuclei in skeletal muscle because it's multinucleated. So this is just an overview of what a single skeletal muscle cell or fiber looks like. Next, let's talk about the striations. So we know striations in skeletal and cardiac muscle are because of repeating arrangements of the myofilaments being actin and myosin. Well, these repeating elements create light and dark bands. So the repeating elements create striations by creating areas where there is overlap of the myofilaments and then areas where there is no overlap of the myofilaments. And if two things are overlapping, their shadow is darker than when there is only one element, so that shadow is lighter, and that creates these dark and light areas. And we can see the dark and light bands in each myofibril along the sarcomeres. So if you have a completely intact muscle fiber, the dark bands, which are also called A bands, and the light bands, which are also called I bands, should be perfectly aligned, and that's what gives the cell a striated appearance. So again, we have dark A bands, light I bands, and that's because of the repetitive arrangement of actin and myosin within each myofibril. So here's a better view of just how many of these rod-shaped proteins called myofibrils you would have in a skeletal muscle cell. So this whole structure of the skeletal muscle cell, it contains many myofibrils, which we know inside of each myofibril are actin and myosin. We can see the units of a myofibril, which are the sarcomeres. And because of the regular arrangement of actin and myosin, the myofilaments, in the sarcomeres of a myofibril, as long as the myofibrils are kind of all lined up together, it creates areas where the overlapping myofilaments produce a dark shadow and areas where the myofilaments don't overlap, which produces a much lighter shadow. So that creates the dark and light bands. So what we've learned is myofibrils are inside skeletal muscle cells. They are densely packed, they are rod-like in structure, and they contain the contractile elements myosin and actin. They make up most of the muscle volume, and therefore they make up roughly 80% of the internal components of a skeletal muscle cell or fiber. And because of the regular arrangement of myofibrils within a muscle fiber or muscle cell, we get perfectly aligned repeating series of dark and light bands, or A and I bands. So now let's look at just a part of a myofibril. So we're going to be looking at just a sarcomere, which are the smallest contractile units of any muscle. You specifically have sarcomeres in skeletal and cardiac muscle, but not in smooth. That's why we don't see striations in smooth muscle. And keep in mind that sarcomeres are just units of a myofibril. So what we see in this image down here is one myofibril, and we see just part of the myofibril, and that is enough to show us a sarcomere, because sarcomeres go from Z disc to Z disc. So earlier I pointed out a zigzag line as the ends of a sarcomere, and those zigzag lines have names. They are called Z discs. Notice that we have thick red lines and thin blue lines. The thick red lines are myosin. The thin blue lines are actin. The A band is a region where the red and blue lines overlap, so that produces the dark A band shadow. The I bands are areas where we only have actin, so there's no overlap, and therefore we have a lighter shadow or a light band. The M line is going to be the area where two thick filaments, or myosin, so this red line, meet, and the H zone is an area where there is no actin. There's just the thick myosin, so there's no overlap in the middle of the A band at the H zone. There's other structures we're going to point out, but keep in mind we're looking at part of a myofibril, just enough to point out a sarcomere. A sarcomere goes from Z disc to Z disc. It's a unit of a myofibril, but also the smallest contractile unit of a muscle. These myofibrils contain actin and myosin, so that makes sense that you'll see actin and myosin within a sarcomere, since it's a unit of a myofibril. The thick filament is another name for myosin, one of our myofilaments. Thin filament is another name for actin, another myofilament. These two myofilaments are the reason that muscle contracts, so these are contractile elements of a myofibril, but specifically of a skeletal muscle cell, because of how many myofibrils you have within one single muscle fiber. Now we've pulled out actin and myosin, you get a better view of what's going on. Again, here would be the dark A band, where we have overlap with actin and myosin. Then you have the light I band in areas where you only have actin, so we don't have as much overlap, less shadow, therefore a light band. M line, where the thick filaments meet in the middle, and then H zone would be this whole area where there is no actin, but it's within the dark band or the A band. So here is just a quick sheet to go over those terms. You can go ahead and pause the video here if you need to look through them, and I'll just read through the terminology if you're listening to this, say in the car. So the A band is a dark band, the I band is a light band, the Z line or Z disc are areas that mark the boundary between adjacent sarcomeres, the M line connects thick filaments but is within the A band, the H zone contains only thick elements in an area within the A band, and the zone of overlap is anywhere where the thick and thin filaments overlap. Let's go back to A versus I band. Yes, the A band is the dark band, but that's because it contains thick and thin filaments. They're overlapping. The I band is the light band because it contains only thin filaments, therefore its shadow is not as dark as the overlapped A band. So let's move forward and look at the anatomy of myosin, and then we'll look at the anatomy of actin. So again, these are the two myofilaments. So we're looking at microscopic structure of the myofilaments within each myofibril, and the myofibrils are within individual cells. So myosin is called a thick filament because it's thicker than actin, the thin filament, and we find that the thick filaments are composed of the protein myosin. Each myosin molecule has a rod-like tail, and usually we have a globular head. You're going to have two tails wrap around each other, meaning that one myosin unit has two globular heads. The myosin globular heads have a binding site for ATP. So what that means is an individual myosin unit contains two globular heads and tails that wrap around each other. On the heads, there is a location for ATP to attach, and that means that there is a binding site for ATP on the globular myosin heads. This binding site is going to be important for creating a cross bridge. A cross bridge is a link between actin and myosin, and it is required for skeletal muscle contraction. Oftentimes, myosin units are going to accumulate meaning that you'll have many tails as the medial portion of the thick filament, and you'll have the globular head sticking out laterally or away from the medial unit. This is actually what the thick filament looks like, so you have many myosin units coming together to create the thick filament. Next, we're looking at the ultrastructure of the thin filament, also known as actin. So myosin and actin are both myofilaments, and the thin filament is slightly thinner than the thick filament. The thin filament, called actin, is made up of actin subunits, just like the myosin thick filament is made up of myosin subunits. However, the actin subunits are completely globular. They're what looks like the little blueberries down here. And there is a binding site for myosin, specifically the globular heads of myosin, on each actin. So this circle that's covered up by this orange line, that is the binding site on actin for myosin. The orange line is a protein called tropomyosin. These yellow structures, kind of globular themselves, are called the troponin complex. They're just globular proteins that accumulate together that makes them a complex. And together, they are called the troponin-tropomyosin complex. Or sometimes they're just called separately as troponin and tropomyosin, and oftentimes they have to move out of the way in order to reveal the myosin binding site on actin. And we're going to hear more about that. So again, the actin subunits are globular. They contain active sites to which myosin heads can bind during a cross-bridge formation, so that's during contraction. The tropomyosin and troponin regulatory subunits must move out of the way in order to allow myosin heads to bind to the actin active site. And right now they're covering up the actin active site that would usually bond to the myosin globular head, and so right now there could be no cross-bridge formation. Alright, so before we go into the T-tubules and sarcoplasmic reticulum, let's review the myofibrils. So inside myofibrils are myofilaments. Myofibrils contain units called sarcomeres. Sarcomeres are the smallest units of contraction. It is because of the repeating units and arrangement of the myofilaments in a myofibril that we see striations. The A band was the dark band, the I band was the light band, and make sure to go back if you need to look at the other terminology associated with sarcomeres. Next, we look at the ultrastructure of myosin and actin. Myosin is also called the thick filament and is made up of myosin subunits. Actin is also called the thin filament and is made up of actin subunits. Myosin heads have a binding site for ATP. Actin has a binding site for myosin heads. But on actin, we see a troponin and tropomyosin structure, and those two proteins are covering up the actin active site that wants to bind to the myosin heads. And unless we move that out of the way, myosin will not bind to actin, therefore that cross bridge that allows for contraction will not form. Alright, let's move on to the T-tubules and sarcoplasmic reticulum. So there's two sets of intracellular tubes that participate in the regulation of muscle contraction. One's the T-tubule, the other is the sarcoplasmic reticulum. Keep in mind that the T-tubule is an extension from the sarcolemma. I cannot point out enough that the T-tubule is connected to the plasma membrane of a muscle cell. I'll even say it again. The plasma membrane of a muscle cell is connected to the T-tubule. That means anything that happens to the sarcolemma, anything that happens to the plasma membrane of a muscle cell can be communicated to the inside of the cell thanks to the T-tubule. So we get an increase in the muscle fiber surface area because of this continuation with the sarcolemma. So T-tubules are extensions from the sarcolemma. They're going to bring electricity or an electrical signal from the outside of the cell to the inside. That's going to be paramount for releasing calcium from the sarcoplasmic reticulum. The sarcoplasmic reticulum is a modified ER or endoplasmic reticulum that abuts the T-tubule via terminal cisternae. So it's a very elaborate smooth endoplasmic reticulum. It surrounds each myofibril, so it's in close contact with the sarcomeres, and it's able to regulate intracellular levels of ionic calcium because it contains calcium. It stores and releases calcium on demand thanks to a couple of signals that happen during muscle fiber contraction. But you must have stimulation of the sarcoplasmic reticulum and calcium in order for contraction to take place. Calcium release allows for the formation of a cross bridge, so calcium provides the final go signal for contraction. That's a very important point. It is calcium that provides the final go signal for contraction. And eventually we're going to really be looking at the physiology. We'll go step by step, how does a muscle contract? And we're going to see these terms show back up. So knowing what they are, where they're located, how to recognize them, is a great way to understand the physiology better. So let's look at them and their location. So we've already mentioned a triad. It's three structures that touch each other, a T-tubule and two terminal cisternae. The terminal cisternae are the parts of the sarcoplasmic reticulum that touch the T-tubule. One touches the T-tubule on the left, one on the right. That creates the triad. And the T-tubules are able to regulate muscle contraction by bringing an electrical signal from the sarcolemma into the sarcoplasm and communicating with the sarcoplasmic reticulum to release calcium. Once calcium is released from the sarcoplasmic reticulum, we see an uptick or an increase in intracellular calcium levels of a skeletal muscle cell. So let's just point out these structures. So we have multiple rod-shaped proteins that contain myofilaments. These are myofibrils. Units of a myofibril are a sarcomere, which goes Z-disc to Z-disc, and it's that regular repeating pattern of myofilaments within a sarcomere that make up myofibrils that give us the A and I band or dark and light band. Around the myofibrils are the T-tubules, here in yellow, and the blue sarcoplasmic reticulum. Notice that parts of the sarcoplasmic reticulum touch the T-tubule. The parts that touch the T-tubule are the terminal cisternae. Then you have the T-tubule in the middle. So if we count 1, 2, 3, 3 parts make up a triad. Important things happen at the triad, so that's why we're pointing out the triad relationship and its structure. Also notice that we can see here that the sarcolemma just folds inwards and becomes a T-tubule. So the T-tubule really and truly is a continuation of the sarcolemma. It's just found within the cell. So the T-tubule is continuous, connected, touches, will communicate. However you need to think of it, know that the T-tubule is continuous with the sarcolemma and can bring a signal from outside the cell to inside the cell. So let's even talk more about that. How do T-tubules regulate contraction? Well, ultimately, we got to start with a nerve-initiated electrical impulse, specifically for skeletal muscle. So we're going to be talking about somatic nervous system control, which uses motor neurons. And motor neurons will communicate with skeletal muscles, and they will have their own electrical signal that they'll then pass on to the sarcolemma of a skeletal muscle fiber. So let me say that in a little easier way. The electrical signal from a motor neuron will travel to the plasma membrane of a muscle cell. And there are steps that allow for this transfer. It doesn't just happen because the electrical signal jumps from one place to another. There's a series of chemical steps that must occur in order to get this electrical signal that originated in a nerve to the plasma membrane of a muscle cell or the sarcolemma. But because the T-tubules are continuous with the sarcolemma, once you get that electrical signal on the sarcolemma, then the signal can travel into the T-tubule because they're connected. We just have electricity in the plasma membrane of a muscle cell. It travels along that plasma membrane to the T-tubule. Then the electrical signal goes into the muscle cell. And again, that's because the T-tubule is continuous with the sarcolemma, continuous with that plasma membrane. Therefore, they can conduct electrical impulses into the cell, into the sarcoplasm, near the myofibrils, which are located next to sarcomeres, which are the smallest units of contraction, because they contain actin and myosin. And if a sarcomere shortens, then the whole myofibril shortens. And if all the myofibrils in a skeletal muscle cell shorten, then the cell shortens. And if the cells of a muscle as a whole shorten, then the muscle as an organ shortens. And it's all because of the T-tubules acting like a rapid telegraph system in order to get that electrical impulse from the sarcolemma to inside the cell close to the myofilaments, actin and myosin. So T-tubules are going to ensure that every myofibril in the muscle fiber contracts at the same time. So they're very important for contraction. Alright, so let's just review the anatomy. We have first our three connective tissue coverings that cover muscle as an organ. The epimysium on the outside, then we have the perimysium that surrounds fascicles, so let's take a closer look at that. The perimysium is going to contain individual skeletal muscle fibers, and around each individual skeletal muscle fiber is its own connective tissue covering, endomysium. Inside a skeletal muscle fiber, or skeletal muscle cell, is myofibrils and other organelles. If we look at a myofibril, we can see that it's closely associated with sarcoplasmic reticulum and T-tubules. T-tubules, again, are continuous with the sarcolemma. We can also see that inside a myofibril are actin and myosin, and we can see a unit of a myofibril, the sarcomere, which is the smallest unit of contraction. We can even take the A and I bands out, or we can take the myofilaments out so that we can see the arrangement of myofilaments that create the dark A band and the light I band. Where we have overlap is the dark A band. Where we don't have overlap is the I band. And this repeating arrangement of myofilaments within the myofibrils, and the myofibrils being lined up in a skeletal muscle cell, is what creates the striations that we see in skeletal muscle. It's also the reason we see these striations in cardiac muscle. And the ends of a sarcomere are Z-line to Z-line, or Z-disc to Z-disc. And make sure to go back and review the terms such as M-line, H-band, and even M-zone. Also keep in mind that the thick filament is another name for myosin, and the thin filament is another name for actin. And so next we're going to be looking at the sliding filament model of contraction. This starts to get into physiology. There's still some anatomy to be talked about, but we're going to start to filter in the why or how muscle contracts with the anatomy. So before going further, make sure you have a really strong understanding of what skeletal muscle anatomy looks like, what the structures are, where they're located, how they're associated, and to a small extent, because we haven't talked about it in detail, what they're for. So make sure to watch the next part of the video.